Hi everyone, I'm Navin Sestra. So today I'm going to present my research that I have done in the last 2 years. So I was associated in mapping the QTL in flavonoids in tomatoes. So I'm going to talk about flavonoids at first. So going with the background in tomato, going with the flavonoids in background. So flavonoids are the secondary metabolites, and they are the polyphenolic compounds, and they are the largest phytonutrients. So till now, 10,000 flavonoids has been identified, and its numbers is still increasing. Since they are the secondary metabolites, they don't play direct role in the growth and development of the plants, and even in the reproduction of the plants. But however, they have a secondary role in the reproduction by attraction of the pollinators, because they are responsible for imparting bright colors in flowers like red color, blue color, and purple color. Furthermore, they are also very useful in the long run, as they play a defensive role against biotic and abiotic stresses. Furthermore, they also have many health beneficial related properties, because they are the antioxidants. So having the antioxidant properties, they are very useful and has been known to reduce the cardiovascular risk factors. They also do have the anti-tumor and anti-cancer properties, similarly anti-osteoporotic properties and anti-inflammatory properties. So they have a lot of health beneficial activities within humans. So now let's go with the evolutionary concept of flavonoids. So in 2002, Close and MacArthur came with a hypothesis called oxidative pressure hypothesis. What does this hypothesis tell us is the oxidative pressure within the plants are increased by many biotic and abiotic factors. The abiotic factors includes excess light, energy, low nutrients, cold and water stress conditions, wherever biotic factors include fungi, bacteria, and herbivores. So whenever these two factors, biotic and abiotic factors, just hits the plant, then the oxidative pressure is increased in the plants, and this oxidative pressure in turn increases the constitutive or induced phenolics within the plants, and these induced phenolics just quenches the reactive oxygen species within the plants and thus reduces the oxidative pressure, balancing the oxidative pressure in the plants. Similarly, the same induced phenolics also reduces the activities of biotic factors and just maintains the oxidative pressure within the plants. So after having this evolutionary concept of flavonoids, let's go with the classification on flavonoids. So before going to the classification of flavonoids, I would just like to start with the polyphenols. So in the polyphenols is divided into three classes. They are phenolic acids, flavonoids, and non-flavonoids. So within these three classes, we are just going to concentrate on the flavonoids. So as I already told you that there are already 10,000 phytonutrients, and out of 10,000 phytonutrients, the flavonoids or the secondary metabolites that are useful for humans are just 6. They are flavonoids, flavonols, isoflavones, and anthocyanidins. Since the flavonoids that are present in tomato is anthocyanidins, so we are just going to focus on anthocyanidins. So now what are anthocyanidins, or what are anthocyanins? Let's go on that. So the anthocyanins are imparted as purple color in tomato, so we love to call it as a purple tomato in case of tomato. So the anthocyanins are the cause to make the tomato purple. So within the anthocyanins, the classes that are responsible for imparting purple color are petunidin, and the trace amount of delphinidin and malvidin are also present. We might be interested that in which area or in which section are those anthocyanins present. So here I want to focus that they are restricted just in skin and are not available on the flesh of the tomato. So since there are already other purple tomato, then why we are just interested to work on the purple tomato that we don't have? Since there are so many heirloom varieties like Black Prince and Purple Cherokee, which look purple, like this in figure, but actually they are not the anthocyanins. They are the pheophytins. So what pheophytins are is, because in those varieties there is a green flesh gene which just prevents the breakdown of chlorophyll and just imparts the brown color and make them purple and make them look purple, but they're actually not the anthocyanins. So we should not get confused with the purple color in those heirloom varieties and the one we are going to make the one in our cultivated varieties. So why we are still interested in tomatoes whenever there are so many fruits and vegetables which have highest concentration of anthocyanins in it. For example, blueberries are the #1 fruits which has highest content of anthocyanins, but we don't have any anthocyanins in the cultivated tomatoes. The reason is, the fact is, so the statistics of 2015 shows that the tomatoes are consumed almost double than that of blueberries. So if we want to look young by consuming the vegetables, then why don't we work on the anthocyanins that are beneficial for us, and that by consuming the tomatoes? So we just want to introduce those anthocyanins in the cultivated tomatoes. So that is the reason that why we are interested in working in anthocyanins in tomatoes. So now let's go with the literature review regarding the anthocyanins in tomatoes. So till now, only the 3 genes have been found responsible for the synthesis of tomato. So they are the anthocyanin fruit, aubergine, and atrovallaceum. So the first 2 genes are available in chromosome 10 and the 3rd gene in the chromosome 7. The first 2 genes are responsible for expressing anthocyanin in fruit, whereas the last one, the atrovallaceum, is responsible for expressing anthocyanin in whole plant. The thing here to be noticed or to know is that the anthocyanin fruit has been introgressed from Solanum chilense, whereas aubergine gene has been introgressed from Solanum lycopersiculates and the atrovallaceum from the Solanum tschmania. Furthermore, the flavonoids in plants, the flavonoids are present in terms of 2 compounds. They are preformed compounds and induced compounds. So the preformed compounds are synthesized by plants in response to physical injury, infection, or stress, whereas the induced compounds are already present in the plants and they are present when they are have to play the direct role in the defense system. So furthermore, the anthocyanins, as I already told you, that anthocyanins has antioxidant activity, which in turn helps to tolerate the heat within the plants. So now let's go with the main research that we are dealing with in our research. So the main research objectives here are the mapping QTL associated with anthocyanin in tomato and assess the role of anthocyanin against early blight in tomato. And the last one is assessing the role of anthocyanin against heat stress tolerance in tomato. So going with the first objective, what we did is we crossed the parents NC74 cap and L-saccharide and generated the BC1 F1 generation, and we backcrossed with L-saccharide and generated BC1 population. So in BC1 population, we quantified the anthocyanin through high performance liquid chromatography method. So now after quantifying the anthocyanin within the BC1 population, we group them into 3 classes based on the anthocyanin content: low, medium, and high. The low groups contain the anthocyanin from 4.3 to 13 nanomole per gram fresh weight. And the medium group, the anthocyanin content of the medium group range from 15 to almost 16 nanomole per gram fresh weight. And of that of high group contain the anthocyanin from 27 to 85 nanomole per gram fresh weight. So within these 3 groups, we just took the lowest and the highest group, and we bulked the DNA from these groups. After bulking the DNA from these groups, we did the whole genome resequencing, applying the next generation sequencing method, and then we aligned those reads to reference sequence. After that, we calculated the SNP index. So here I just want to focus what does SNP index means. So the SNP index is calculated by dividing the alternate allele depth by total read depth. So what does alternate allele depth and total read depth means is, so here in figure we can see the reference sequence in the orange color, and here we can see in the horizontal, in the vertical lines, there are 10 reads. So that is the total read depth, and the alternate allele depth is, so if the sequence of the low group or high group. So the first one, let us take the example that the first one refers to the sequence of low bulk, and the second one refers to the sequence of high bulk. So if we just regard them as a low bulk and high bulk, the first one we can see that almost 4 of the alleles are different than that of the reference sequence. So here the alternate allele depth comes to be 4 divided by 10, that is 0.4. However, in the next, what we consider as a high bulk, there we can see almost 10 of the alleles are different than that of the reference sequence. So here the alternate allele depth is 10, and when we divide that by total read depth, that is 10, that comes to be 1. So this is the way that how we calculate the SNP index. So after calculating SNP index, we filtered the false SNPs. So the SNPs which are less than 0.3 or the depth of less than 7 times. So whenever we got the SNP index less than 0 and read depth less than 7, then we called it as false SNPs and we removed it from our data. So after calling the SNPs, we calculated the del SNP index. So what del SNP index is, it is the difference of SNP index between high bulk and low bulk. So after calculating the SNP index, we calculated the average del SNP index by sliding window. It is a software which generates the QTL analysis plots. So this is the QTL analysis plot generated by the sliding window. So here in this figure, we can see a curve that is generated by the sliding window based on the number of SNPs available within that chromosome that we sequence through next generation sequencing method. So here the red line indicates the 95% confidence intervals. So as I talked to you about the del SNP index of high bulk and low bulk, so the graph that we can see, the SNP index of low bulk looks like this, and that of high bulk looks like this. So now we just took the difference of these two bulks and we got these graphs. After having this graph, so we calculate the absolute SNP index. After calculating the absolute SNP index, we just took the sum of the absolute del SNP index, and this is the result that what we got through QTL-seq approach. So here in this last graph, we can see that chromosome 1, chromosome 2, chromosome 4, and chromosome 7 has the peaks that crosses the 95 confidence intervals. So we can say that the QTLs responsible for the synthesis of anthocyanin are present in chromosome 1, 2, 4, and 7. But however, the QTLs in chromosome 7 has already been reported. So we want to say that we were able to discover the novel genes or novel QTLs in chromosome 1, 4, 2, and 4. So our conclusion is that we found 4 QTLs. 1 QTLs in chromosome 1, which started at 9.96 MB and ended at 11.54. The total length is 1.58. And in chromosome 2, the QTL was located in the region of 46.94 to 51.88. And that in chromosome 4, we found 2 QTLs. The first QTLs was located in the region between 8.31 to 11.9 MB. And in chromosome 4, it was located in between 15 to 20 MB. So these are the 4 novel QTLs that we found in our research. So this is the thing that we have done in 2 years to find the QTLs associated with anthocyanin in tomato. So if you have any queries or any questions regarding this approach, del SNP index, how we calculated that, and about the QTL-seq method, then please feel free to send me an email or through any communication means so that you can get to me to get those answers. Thank you for your time and for listening to my video. Thank you.